The main advantage of this plate-based turbidity assay is that it is quick and allows for several amyloid beta fibrogen interaction inhibitors to be screened at once without sophisticated setup or requirements.
The hit compounds identified in the turbidity assay can be further validated for their ability to restore a beta-induced structural abnormalities in the fibrin clots as assessed by scanning electron microscopy.
Visual demonstration of clot preparation for scanning electron microscopy analysis is critical, as any variations in the preparation can contribute to variations within the experimental results.
The focus of this demonstration here is on A-beta fibrinogen interactions. This protocol can be readily modified to analyze other interactions with other proteins and compounds with the fibrin clot.
Begin by adding 1.5 micromolar of freshly prepared fibrinogen in 200 microliters of clot formation buffer to each A-beta -42 containing and control wells, and incubate the plate on a rotating platform at room temperature. After 30 minutes, simultaneously add 30 microliters of freshly prepared thrombin solution directly into the center of each fibrinogen-containing well to initiate clot formation, and immediately read the absorbance of the in vitro clots at 350 nanometers, repeating the measurement every 30 to 60 seconds over the course of 10 minutes. To evaluate the effect of A-beta fibrinogen interaction inhibitors on fibrin clot structure, first use forceps to place clean 12 mm siliconized glass circle cover slips into